Welcome to another episode from the Veterinary Medicine Digest. It starts with a simple meal, a few mouthfuls of hay in a quiet pasture. The sun is setting over a ranch in Oregon, and the air is cool and damp. The horses are grazing, moving slowly from one bale to the next. It looks like a scene of pure tranquility. But hidden within that hay is an invisible enemy. It is a killer that has been waiting for this exact moment. A killer that does not need to breathe, does not need light, and can survive heat that would boil water. It is a biological machine of terrifying precision. And once it enters the bloodstream, the clock starts ticking. Within days, these powerful 1,000-pound animals will be unable to lift their heads. They will be unable to swallow. And eventually they will be unable to draw breath. This is not just a disease, it is a physiological catastrophe. This is the story of equine botulism. To understand this killer, we have to go back to the late 19th century. In 1895, in a small village in Belgium, a group of musicians gathered for a funeral wake. They ate raw salted ham. Within days, dozens were paralyzed. 3 died. A scientist named Emil van Ermingen was called in to investigate. From the spleen of one of the victims and from that contaminated ham, he isolated a bacterium. He called it Bacillus botulinus. We know it today as Clostridium botulinum. It is a name that strikes fear into public health officials and veterinarians alike. Because what van Ermingen discovered was not just a germ, he discovered the source of the most poisonous substance known to mankind. Think about that for a second. We talk about cyanide. We talk about arsenic, we talk about the venom of a king cobra, but botulinum neurotoxin is on a different level entirely. To put it in perspective, the median lethal dose for arsenic is roughly 15 milligrams per kilogram of body weight, but for botulinum neurotoxin, the dose is measured in nanograms. A single gram of crystalline toxin, if it were evenly dispersed and inhaled, could kill more than 1 million people. It's so potent that it is classified as a category of priority agent for bioterrorism, alongside anthrax and the plague. It has been weaponized by nations. It has been used by cults in attacks on military bases in Japan. But for the horse, this weapon is not found in a missile. It is found in the soil. It is found in the grass. It is found in the very food they need to survive. The organism itself, Clostridium botulinum, is a gram-positive, spore-forming obligate anaerobe. That last part is key. It is an obligate anaerobe, meaning it can only grow where there is no oxygen. It is ubiquitous. It is everywhere. It is in the soil beneath our feet and in the sediment of our lakes. But most of the time it is dormant. When conditions are harsh, when there is too much oxygen or not enough food, the bacterium transforms. It creates a spore. These spores are like tiny armored bunkers. They are characteristically described as drumstick-shaped. They can withstand intense heat, they can survive boiling, they are resistant to radiation and most disinfectants. They can sit in the soil for years waiting. But when the environment becomes perfect, the spores wake up, they germinate. They need an anaerobic environment, a neutral or slightly alkaline acidity level, and a source of nutrients like decaying organic matter. This is why we see outbreaks in horses associated with spoiled haylage or silage. If a bale of hay is wrapped in plastic while it is still too wet, and the acidity does not drop below 4.5, it becomes an incubator, it becomes a factory for the toxin. And that toxin is what does the killing. Now let's get into the science, because at a PhD level, the way this toxin works is nothing short of surgical. The botulinum neurotoxin is a single polypeptide chain with a molecular weight of 150 kilodaltons. When it is released, it is cleaved by proteases into 2 parts, a heavy chain of 100 kilodaltons and a light chain of 50 kilodaltons. These 2 chains are held together by a single disulfide bond. This structure is roughly 35% homologous with the toxin that causes tetanus, known as tetanospasmin. But while they look similar, they act in completely opposite ways. Tetanus causes spastic, rigid paralysis. Botulism causes flaccid, limp paralysis. The heavy chain is the delivery vehicle. Its job is to find a very specific target, the peripheral cholinergic nerve terminals at the neuromuscular junction. It does not go to the brain. It cannot cross the blood-brain barrier. It stays in the periphery. The carboxy terminus of the heavy chain binds with exquisite affinity to receptors on the presynaptic nerve membrane. We believe this involves a double interaction with both a protein receptor and acidic lipid gangliosides like ganglioside D1A. Once it is bound, the nerve cell is tricked. It takes the toxin inside through a process called receptor-mediated endocytosis. Inside the nerve terminal, the toxin is trapped in a small bubble called an endosome, but the environment inside that bubble starts to change. It becomes acidic. This drop in potential hydrogen triggers a structural change in the toxin. The heavy chain forms a pore in the membrane of the bubble, allowing the light chain to slip through into the cytosol of the nerve cell, and this is where the real damage happens. The light chain is a zinc-dependent endopeptidase. It is a molecular pair of scissors. Its target is a group of proteins called the soluble N-ethylmalamide sensitive factor attachment protein receptor complex, or the SNARE complex. These proteins are the essential machinery of life for a nerve cell. They are what allow the nerve to talk to the muscle. Normally, when a nerve signal arrives, little vesicles filled with a neurotransmitter called acetylcholine move to the edge of the cell. The SNARE proteins act like a docking system. They pull the vesicle tight against the cell membrane until they fuse, and the acetylcholine is released into the gap between the nerve and the muscle. The muscle senses the acetylcholine and contracts. But the botulinum light chain cuts those SNARE proteins. Different types of botulism target different proteins. Type A and Type B cleave the synaptosomal-associated protein of 25 kilodaltons. Type B, Type D, Type F, and Type G target synaptobrevin, also known as vesicle-associated membrane protein 2. Type C is even more destructive. It cleaves both the synaptosomal-associated protein and another protein called syntaxin. Regardless of the type, the result is the same. The docking station is destroyed. The vesicles are still there, full of acetylcholine, but they cannot dock. They cannot fuse. They cannot release their message. The nerve signal arrives at the terminal, but the muscle never hears it. The connection is broken. The muscle goes limp. And it stays limp until the body can grow entirely new nerve terminals, a process that can take weeks or even months. In horses we see 4 main serotypes, A, B, C, and D. There is a distinct geography to this. If you are in the mid-Atlantic United States or Kentucky, you are dealing with Type B. It is responsible for more than 85% of cases in North America. It loves the soil there. It loves the round bales and the wet hay. But if you move west of the Rocky Mountains, the landscape changes. Now you are in Type A territory, and Type A is a different beast. A decade-long review of cases from 1998 to 2008, conducted by the Botulism Laboratory at the University of Pennsylvania, paints a grim picture of Type A. Almost all of these outbreaks occurred in the western United States, with Oregon and Idaho being hit the hardest. These were not just isolated cases. These were mass casualties. In one outbreak in Oregon, 40 horses were exposed to bluegrass hay that had been spread along a creek. The weather was unusually warm and wet. The hay became matted and damp. 20 horses died within the first 3 days. 4 more died over the following week. That is a 60% mortality rate in a single group. This highlights the 3 distinct ways a horse can be poisoned. The first is the most common in adults, ingestion of preformed toxin. This is what we call forage poisoning. The bacteria grew in the hay, they produced the toxin, and the horse simply swallowed it. The second route is toxico-infectious botulism. This is the nightmare of the breeding farm, also known as shaker foal syndrome. This happens most often in foals between 2 and 4 weeks of age. Their gastrointestinal tracts are immature. Their normal microflora is not yet established. They swallow the spores from the soil, and instead of just passing through, those spores germinate inside the foal's gut. The bacteria set up shop and start pumping out toxin from within. The third route is wound botulism, which is rare but deadly. This happens when a wound, like an umbilical infection in a foal, or a site of a surgical procedure like a castration, becomes contaminated with spores. If the wound is deep and anaerobic, the bacteria thrive. And finally, there is iatrogenic botulism, where we, as veterinarians, accidentally cause the disease. Botulinum toxin is actually used as a medicine to treat things like laminitis or certain muscle contractures. But because horses are so sensitive, estimated to be up to 10,000 times more sensitive than mice, the therapeutic window is incredibly narrow. Even a slightly too high dose can be fatal. So how do you recognize this when you see it? Because the signs can be subtle at first. A horse might just seem a little depressed. Maybe it is taking a little longer to finish its grain. An owner might think it is just a mild case of colic, but look closer. One of the earliest signs is a loss of muscle tone in the tongue. There is a simple field test for this. You reach into the horse's mouth, grasp the tongue, and pull it out to the side. A normal horse will pull it back in instantly. A horse with botulism will let it hang there, or will only be able to retract it slowly and partially. Then comes the dysphagia, the inability to swallow. You might see the horse submerged its muzzle in the water bucket, trying desperately to drink, but the water just splashes back out. You might see saliva or green-tinged fluid running from the nostrils. Then the weakness spreads to the eyes. The pupils become dilated and sluggish. The eyelids start to droop, the tail tone disappears. The horse starts to shuffle its gait, dragging its toes, struggling to hold up its own weight. It starts to tremble, those classic muscle fasciculations in the triceps and the gluteals, as the muscles exhaust themselves just trying to keep the animal standing. Eventually, the horse goes down. And once a horse is recumbent, the prognosis becomes extremely guarded. In adult horses, if they are down for more than 24 hours, the survival rate drops to 20% or less. The massive weight of their own bodies starts to work against them. They develop decubital ulcers. They get compartment syndrome in their muscles. They develop aspiration pneumonia because they can't protect their airway, but the ultimate cause of death is usually respiratory failure. The diaphragm, the muscle that pulls air into the lungs, eventually loses its nerve connection. The horse slowly suffocates. Diagnosing this is one of the greatest challenges in equine medicine. It is often called a diagnosis of exclusion. Why? Because the amount of toxin it takes to kill a horse is so small that we can almost never find it in their blood. By the time the horse is showing signs, the toxin has already left the circulation and is bound inside the nerve cells. It is gone. It is invisible. For decades, the gold standard has been the mouse protection bioassay. It is a grueling test. You take a sample of the horse's serum or feces or the suspected feed. You inject it into the abdomen of a laboratory mouse. Then you wait. You watch for the classic signs in the mouse, a laboured breathing pattern that gives them a wasp waist appearance, followed by paralysis and death. To prove it is botulism, you take another group of mice, give them the same sample, but also give them an antitoxin. If those mice live, you have your answer. But there are major problems with this. First, it takes 4 days to get a final result. A horse with botulism doesn't have 4 days. Second, it is not very sensitive for horses. Because horses are so much more sensitive than mice, a horse can be dying from a concentration of toxin that the mouse bioassay can't even detect. It is estimated that the mouse bioassay only catches 32% of adult equine cases. We can improve that by culturing the feces to look for spores, but that takes even longer. This is why researchers are racing to develop new diagnostic tools. We are looking at enzyme-linked immunosorbent assays that use antibodies to find the toxin. We are looking at quantitative real-time polymerase chain reaction to find the genes of the bacteria. There are even experimental biosensors using fiber optics and gold nanoparticles. Some of these can detect the toxin at femtomolar or even attomolar concentrations. We are talking about finding a single grain of sand in an entire desert. But for now, in the field, the diagnosis remains a clinical one. If it looks like botulism, you have to treat it like botulism. Immediately, the treatment is a race against time. The single most important thing you can do is give the antitoxin. The antitoxin consists of antibodies that circulate in the blood and bind to any free toxin before it can reach the nerves. But here is the catch. The antitoxin only works on the toxin that is still in the blood. It cannot cross into the nerve cell. It cannot undo the damage that has already been done. It can't reconnect the snare proteins. This is why we often see horses continue to get worse for 24 to 48 hours even after they get the antitoxin. We are just stopping the progression. We aren't reversing the paralysis. In the United States, we have a monovalent antitoxin for type B, which is relatively affordable. But if you are in the West and you suspect type A, you need a polyvalent antitoxin, and it is expensive. We are talking $2,500 for a single dose. In a large outbreak, the cost can be staggering, and the nursing care is even more intense. We have to keep these horses in a quiet, dark, padded stall. Any excitement or stress makes them use up their remaining acetylcholine faster, which hastens the paralysis. We have to feed them through nasogastric tubes. We have to manually empty their bladders. We have to turn them every few hours to prevent pressure sores. For foals, the story is a bit more hopeful. They are smaller, they are easier to manage. We can actually put them on mechanical ventilators to breathe for them while their nerves recover. We have seen survival rates in treated foals rise to over 90%. But for adult horse, a ventilator is almost impossible. The logistical and financial costs are too high, and the complications are too many. So if treatment is so difficult, prevention becomes everything. And the good news is that we have an incredibly effective vaccine for type B. The American Association of Equine Practitioners recommends it as a risk-based vaccine. If you are in an endemic area, you use it. It is a 3-dose series, followed by an annual booster. Pregnant mares are vaccinated 4 to 6 weeks before foaling to pass those life-saving antibodies to the foal through the colostrum. But there is a major gap in our defences. That vaccine only works for type B. There is no commercially available vaccine for type A or type C in the United States. If you are a rancher in Oregon or Idaho, you can't just vaccinate your way out of this. You have to manage the environment. You have to be obsessive about the quality of your pay. You have to keep it dry. You have to keep it off the ground. You have to watch for carrion, like a dead rodent that might have been caught in the baler. A single decaying carcass can contaminate an entire batch of feed with type C toxin. As we look to the future, the research is focused on filling these gaps. We are seeing the development of recombinant vaccines that use only a piece of the toxin's heavy chain. These are safer to produce and can provide protection with a single dose. We are looking at new treatments like small molecules that can actually enter the nerve cell and inhibit the toxin's enzyme activity, potentially reversing the paralysis. But for now, equine botulism remains one of the most daunting challenges a veterinarian will ever face. It is a reminder of the incredible power of the microbial world. It is a reminder that the most dangerous weapon on Earth isn't something we built in a lab. It is something that has been living in the soil for millions of years, just waiting for a wet bale of hay and a hungry horse. It is a scandal of nature, a silent, invisible executioner that leaves no trace and takes no prisoners. Understanding the molecular precision of the botulinum neurotoxin doesn't just help us save horses, it helps us understand the fundamental way that all life communicates. Every time you move your hand, every time you take a breath, you are using the same snare proteins that this toxin destroys. By studying this killer, we are learning how to protect ourselves, not just from disease, but from the potential misuse of these toxins as weapons. It is an interdisciplinary battle involving microbiologists, neurologists, and clinicians. In the end, the story of equine botulism is a story of vigilance. It is about the rancher who notices a slightly matted bale of hay and decides not to feed it. It is about the veterinarian who sees a horse drooling and immediately checks its tongue tone. It is about the researcher spent late nights over an enzyme-linked immunosorbent assay looking for a better way to find the invisible. It is a fight won in the details. Because when you are dealing with the most poisonous substance on Earth, there is no room for error. The outbreaks in the West, the Shaker foals in the East, the historical shadow of biological warfare, they all point to the same truth. We live in a world shared with giants and with invisible machines. Our task is to stay one step ahead of the armor-plated spores and the zinc-dependent scissors that want to cut the cord between the mind and the muscle. It is a deep dive into the very fabric of physiology. And it is a dive we must continue to take, for the sake of every horse in every pasture, waiting for their next meal. Thank you for listening. The titles for the reference papers are in the video description. From VetMed Digest, where we keep minds inspired, hearts light, and tails wagging.